Hello and welcome, Michigan soybean farmers. My name is Jana Fritz. I'm the Chief Executive Officer of the Michigan Soybean Committee and the Michigan Soybean Association. Thank you for joining us today for our virtual winter meeting series. I'm coming to you today from our family farm. We live and farm in the thumb of Michigan in the Pigeon, Elkton, Bad Axe area. We operate 1,500 acres with soybeans, corn, wheat, and dry edible beans. And we are excited to also learn about all of this agronomic research that will be provided to you today during this winter meeting series. I wanted to take just a minute and introduce you to some of the things that your soybean checkoff is working on. First, let me also say thank you to you, the farmers, for supporting the Michigan Soybean Checkoff. Your dollars from the checkoff come directly to our organization and help facilitate three main focus areas. Those main focus areas include, of course, production research, which is why we're here today, market development, and outreach activities. To name a few examples, other than today's agronomic research meeting, some other focus areas for the production team include seed commercialization and our request for proposal research that we conduct annually with Michigan State University and other collaborating research partners. Market development activities are centered around things like growing demand for biodiesel, finding new emerging international markets for soybeans, and working to research new alternative uses for soybeans and soybean oil. Finally, outreach efforts include consumer education, school children education through our Soybeans Go to School kits, as well as farmer education in many facets, whether that be some of our other focus areas through production or market development as well. We want to make sure you, the Michigan farmer, are as up to date on all things that you need to know about your soybean industry. As if those focus areas weren't enough, we also partner with the Michigan Soybean Association, who takes up the charge through their lobbying and advocacy efforts. This is a separate organization from the checkoff. They are allowed to lobby and advocate in the Michigan and national legislatures for soybean-related issues. Our partnership between the checkoff and the association is critical to the overall influence and expansion of the soybean industry in Michigan. And we need both arms of the organization to move forward for the future. Thank you again for coming and participating in this meeting. We, of course, are so sad that it has to be virtual again this year, but we will look forward to the day where we can be together again in person. As we move forward in this meeting, allow me now to introduce Lori Eisley, our president of the Michigan Soybean Committee.
Thank you, Jana. It is my pleasure to welcome each of you to these winter virtual meetings. I hope that the information that you gain from our on-farm research is something that you find valuable. I certainly hope that it's something that you can put into practice on your own farms. This program is one we're especially proud of because it does take place on Michigan farms by Michigan farmers, and they're very large-scale projects. So a little different than what we do at the university, where we might be doing things in the greenhouse or on a really small plot. I know my husband and I farm in southeastern Michigan, and we gained, especially from the harvest equipment workshop that was put on, using some of these funds in making the decision to change to a draper head. And certainly other areas that we've implemented some of this information have been in looking at earlier planting dates and how that might impact our yields. So I hope that you also will be able to take some of this information and find it valuable in your own operation. We're also always looking for new ideas for things to do for on-farm research. So I hope if something comes into your head that you'd like to share, that you will pass that on to the people doing these presentations. And wishing you all a safe and healthy winter.
Thank you, Lori. We will turn it now back over to Mark and Mike for your agronomic on-farm research. Thank you.
Hi, I'm Mark Seaman, the research manager with the Michigan Soybean Committee. Our goal is to make the best of this virtual meeting format. Most presentations have been recorded, but the speakers will be available live to answer questions. To ask those questions, please use the chat function on the top or bottom of your screen to type your question, or click on the raise hand icon to be called on to ask your questions after you've been unmuted. Please stay tuned in until the end of this session to provide your input on trial directions for this year in 2021. Our main presenters today are Dr. Marty Chilvers, the MSU Field Crops Plant Pathologist, and Mike Staton, the MSU Extension Soybean Production Educator. Enjoy.
Well, I'm Mike Staton. I'm a soybean educator with Michigan State University Extension. My position is jointly funded by your Michigan Soybean Checkoff and MSU Extension. I get to focus 100% of my time on soybean production issues. And I want to welcome you to this presentation where Dr. Marty Chilvers and I are going to jointly present because we have some synergy between our on-farm trials and Marty's small plot trials on campus. So Marty, if you'll tell us a little bit about yourself.
Yeah, just give you a quick update for those that don't know me. So I'm the MSU Field Crop Pathologist. So I run a program based on campus. We work around the state looking at field crop disease management with a pretty heavy focus on soybeans.
Really appreciate the work that you do on soybeans, Marty. It's excellent, excellent work.
Thanks.
We're just going to go ahead and jump into some of these slides and talk about our on-farm research, and then Marty will show his small plot research, and we'll tie things together and answer any questions or comments that the audience has. Before I do that, I want to put up this slide because it's really important. It's a lot of words, but basically the words boil down to an important concept. Everything that MSU does and everything that MSU Extension does is available to everyone. So that's the concept of this slide, and I think we would all agree with that. This is a kind of important slide because it shows the 13 on-farm trials that we did with the on-farm soybean research program. And the ones that are bolded are the ones that Marty and I are going to tag team on today. The others are more agronomic in nature, and we're not going to talk about those. But we will talk about the ones that are bolded. So I'll just jump right into this first one. It's probably our second strongest database of our on-farm trials. We've been doing these base seed treatment trials for the last 4 years, and we have a total of 31 locations over 4 years. And you can see we have pretty good geographic distribution. What we did was kind of interesting is we didn't have a set base seed treatment. What we did is we let the producers choose whatever they were normally applying on their seed. And then we had the same seed lot divided with naked seed and the base seed treatment. And we applied that on 31 locations. And the other thing we did is we tried to game the system. We did ask the producers to put these trials in fields where they had difficult conditions in the past, either emergence problems or poor stands or just a bunch of different things. So that's what we tried to do with these trials. This is a picture of one of our trials up in the St. Charles area. And what you see here is to the right of the flag is going to be 6 rows of treated. Over here to the left of the flag is going to be 6 rows of untreated. And there is a visible difference there. The question is, does that visible difference translate into an economic yield? This is one of the diseases that we use seed treatments for. This is Phytophthora Root Rot. And seed treatments are very effective in managing this disease. They're one of the tactics. And this is just a close-up of affected plants. So here's our yield data. This is 31 sites over 4 years. And the way this is presented is this line right here is going to be the naked seed. That's the untreated. And the height of the bar is going to be the response that we received from the seed treatment. So starting here at the left, we had actually a 10.1 bushel advantage at this one site. It is an outlier, but it was real. It was definitely real. It occurred in almost every rep in 6 replications. On the opposite end of the spectrum, though, we have a situation down here. We actually had a statistically significant yield decrease. If the numbers are not bolded, we can't really say that the effect is due to the treatment. So it's only the bolded ones that you can really focus on. And that was a little disturbing down here in Cass County. What we think happened is that the seed just didn't flow through the planter as well. The treated seed didn't, and we drastically underplanted is what we think happened at that site. So when you average all of these sites together, well, let me explain one other thing about this chart. You'll always see a bar going across here, and that's the break even. So You can sort of tell at a glance which one of the trials made money and then which ones of them didn't, just at a glance. So that's why we do that. When we averaged all of our 31 locations together, it was a slightly profitable practice. We needed 1.3 bushels per acre to pay for it, and we ended up with 1.4. So that's kind of the message, is it's kind of a mixed bag. We had about 10 trials that did give us a statistically significant yield increase and profitable, but the rest of them did not. It does affect stand. We have found that soybean seed treatments do affect stand. And if we look at just this year, we only took the stand counts at the Saginaw site, and it bought us about 10,000 plants per acre. So that is significant. If you can reduce your planting rate by maybe 10,000 plants per acre, that's helpful. So it did buy us that much in that location. Now, when we average it across all the sites, though, we did not see that big of an increase. We ended up with more like 5,000 plants per acre, but there was still statistically significant. The treatments did improve stands for us. Marty, you've also looked at seed treatments, and could you talk about what you've seen?
Yeah, we look at seed treatments every year. Sometimes it's very targeted, so we will develop a field with very high rhizoctonia pressure or something like this, and then test some typically new seed treatments for companies. But a couple of years ago now, we did some work using the standard chemistries that were available at the time, and looked at them across the state. So we had 7 locations using the soybean performance trial locations. We used 2 Asgrow varieties and 2 Pioneer varieties and placed them at these 7 locations and looked at them for 3 years. We had different levels of seed treatment too. So this is asking that extra question: do I need that insecticide? Do I need that nematicide? What is that fungicide component doing for me? So we had the untreated, the fungicide only, fungicide plus insecticide, and then what we would consider a full package. So typically that's like the Poncho Votivo mix. In a nutshell, we found very similar sorts of results to what Mike was talking about. Variable responses, right? So we've got the different counties here just abbreviated to 2 letters. You can see in the middle of that plot, the Allegan. That was a location where we saw some very positive responses. And pretty much every year we did the trials, we saw a positive response at Allegan. So there's something unique about that particular farm where we tend to see very positive response to seed treatments. And so what we're looking at here is the yield relative to the untreated or the naked seed. Okay, so lines above the 0 is a positive yield response, and those that are below a negative yield response. And we saw some of these negative yield responses. They weren't always statistically different, but very similar to what Mike was talking about. Sometimes we see a negative impact, right? This is, it's a little bit difficult to sort of graphically represent all of the data points that we had. But essentially what we were showing is that the prophylactic use of seed treatment, so you just applying seed treatment to everything, was not always economical, right? Very similar to what the data that the on-farm trial data that Mike was showing. I think the numbers pretty much agree. It's right at the cusp of being economically viable on average, but not always. And so it sort of makes you question, when should we be applying these things? There's a lot of other factors that go into these decisions. We conducted these trials at 160,000 seeds per acre, which I would consider a moderate, maybe to a higher sort of seeding rate these days. The locations, again, are very different. Again, Allegan was very, very responsive. Other locations were less responsive. And so that sort of plays into the decision whether to use a seed treatment or not. Having said all of that, it certainly reduces risk, right? So the seed treatment reduces risk by maintaining plant stand, especially at those lower seeding rates. So if we're trying to cut production costs by reducing seeding rate, it becomes more important to use a seed treatment. And especially at earlier planting situations where the seedlings are going to go through more stress. It's more important to protect them from those stresses, those environmental stresses and disease stresses. And so basically reiterating, the take homes from this, and Mike, feel free to chime in here, certain locations we see an amazing return on investment, right? And it was pretty, Mike, you had some a location for the last 3 years now, I think you were seeing around about a 10 bushel, 8 to 10 bushel response rate. So unfortunately, we don't know how to predict that yet, but that is some work we are trying to, or some things we are trying to achieve. We want to be able to take soil samples and predict where you should use a seed treatment for maximum return. There are a number of factors that sort of go into to why that happens. So that's something we want to get a handle on. And just to reiterate, you know, the chance of return on investment greatly increases when we're planting early and when we're at lower planting populations. Mike, do you have any other comments on that?
The only comment I would make, Marty, is what about genetic resistance or tolerance? Is that going to impact the opportunity for maybe getting a better return from our seed treatment if we have a more susceptible variety versus a more tolerant variety?
Yes, I would say so. That's going to factor into it. I would be careful, I guess, about relying on the catalogs, because you know the companies do the best they can in terms of rating and ranking material. But yes, that can certainly play into it. I don't think we've seen a clear separation in terms of susceptible and resistant that really helps us to make that decision. And I think really it comes back to the location in that particular operation. Do you typically see a response from that seed treatment? That's a really good question. Maybe something we can take question and answer from at the end of this session.
Sounds real good. I just want to also echo again what Marty said about the location effect is just huge. This field in Saginaw County 3 years in a row, so it showed a positive response to seed treatments, even though the seed treatments and the varieties did change from year to year. So something about the site is there. We had another site down in Ionia County that very consistently, the person actually replicated all the way across an entire 80-acre field. And every single strip basically showed a response to the seed treatment. So it is a location specific effect is what we're seeing with our strip trials as well.
And I guess the only other economic piece to this too is when you do, typically, I guess when you do pay for that, the full seed treatment, there comes some sort of assurance or insurance with any replant costs, right? And so that's something that's a little bit hard for us to factor in here to this economic analysis, but it's a component, right, of the decision-making process.
Yes.
Yeah.
Yep.
Okay. So let's talk about Phytophthora root rot for a second. And this certainly relates to seed treatment as well. Mike showed you a couple of pictures there. So basically, the Phytophthora, come June, July, you may see a lesion starting to move up the side of the soybean stem, perhaps killing the plants like this. And typically that's going to be Phytophthora. Certainly, get a sample into the diagnostic clinic to get that confirmed. I think it's always important to know what you're dealing with in the field so you can make correct management decisions for next season. So the student here did a whole bunch of collecting Phytophthora isolates from across Michigan. So we solicited a whole bunch of soil samples. He went through this. I just put this slide up to show you it's not a simple, get the soil, put it in a machine, and we know the answer, right? This does take about 8 months to go through the whole process, unfortunately. So we get soil in, we go through an incubation period, we put soybean seed into that soil. We use little cups here, and we bait out Phytophthora isolates from plants that get disease. And we end up in that bottom left picture there, a pure isolate of Phytophthora sojei that we can then ask questions about it. What's its fungicide sensitivity? Which resistance genes is that pathogen overcoming? So next slide. And then, so one of the questions we were answering is, what is the race of Phytophthora sojei, or the pathotype? Pathotype's a sort of standard word that we use. And so essentially we're trying to figure out which resistance genes are still working and which ones are failing against these different Phytophthora isolates. And so we use a needle there and we mark up the plant and inoculate. And this is a cartoon, and you can see Austin there in the greenhouse actually doing the inoculations with a syringe and pushing in the Phytophthora isolate onto the outside of the plant to see if it'll kill it or not. And so this cartoon shows you the different soybean lines that we have. And each of these lines has a single resistance gene to Phytophthora. And you can see in this cartoon, we're essentially showing you, for example, Mike's got his cursor on RPS 1K. That's that's okay. That's a heavily used source of resistance at the moment. For this particular isolate, a single isolate of Phytophthora, when we put it onto that soybean plant, it killed that plant. So in this particular cartoon, we were finding that only the resistance gene 3A, 3C, and 4 were effective at stopping this particular isolate from killing the soybean plants. So in a field situation, that 1K wouldn't be working for us. We would need to be using 3A, 3C, or 4 as a resistance gene. And so essentially what we did, a survey like this was done about 20 years ago now, published in 2001, and that's shown there in the blue lines. And so what we're showing you here is the percentage of Phytophthora isolates that are out there in Michigan that cause disease on these different resistance genes that we have. So in blue there is the 20-year-old data, and you can see there that, for example, RPS1B, we've only got about 25% of the isolates causing disease on 25% of the isolates causing disease on that resistance gene. RPS1K is also pretty effective. You know, it controls most of the isolates. There's only a few that cause disease, 3A, 3B, etc. They put that dashed line. That dashed line is sort of a line that we typically use to determine if that gene is effective or not. It's somewhat arbitrary, but that's the line we used. So the point of this slide, though, is that when we came back in 2020 and repeated the survey, those genes that were effective 20 years ago are no longer effective. And so you can see, for example, RPS1B, we went from only a few of the isolates causing disease to nearly 90% of the isolates capable of causing disease on that particular resistance gene. And same for the RPS1K, which is a heavily relied upon gene. RPS1K and 1C are pretty widely deployed. I think they're in something like 90% of the germplasm that's out there. I believe we have some access to 3A in some of the germplasm or some of the varieties, but it's a small percentage, something like 5%. So the point is, relying on these resistance genes is not necessarily going to work for us, right? And so the seed companies are going to have to try and incorporate some of these different resistance genes if they want to rely on that type of resistance for Phytophthora management. So the action points here is that some RPS genes are becoming less effective, right? It might say on the bag, you've got resistance to this particular race of Phytophthora. Well, that may no longer be the case, right? Because we've got different races of Phytophthora developing. And so the point here is, in terms of the action steps for you guys, if you've been dealing with Phytophthora, make sure you select varieties with a good field tolerance score. So Mike was talking about that earlier. Try and select those varieties that have good emergence, good field tolerance score. And again, speak to your seed dealer, right? They should have a little bit more information, perhaps more so than what's available in the catalog. And then additionally, it really emphasizes the importance of an effective seed treatment. And this is something else we're looking at, the sensitivity. We're checking for resistance to fungicides, right? So far, we have not seen any resistance to fungicides. That's really good news. Typically, metalaxyl mefenoxam have been around for about 40 years as a seed treatment option, and they're pretty much included in every seed treatment that is out there, every formulation. There's a couple of new treatments coming through, Lumicina from Corteva and Intego from Valent. And they also provide really good management of Phytophthora. So we may be pairing some of these newer treatments with that metalaxyl mefenoxam to improve our resistance or improve our management, sorry, of Phytophthora out in the field. So any comments on that, Mike? Did I explain that okay?
Marty, really excellent. My understanding is that the field tolerance doesn't kick in immediately after planting, that it kicks in after the plant emerges or up to some stage the plant is vulnerable. So we really do need those seed treatments if we're relying on field tolerance.
That's right. That's a really good point. So yeah, field tolerance is a good score. Some of that type of resistance, yeah, doesn't kick in until the plant, so like at a V4 growth stage or V2, something like that. They don't necessarily activate at that seedling stage. So that's why it's important to pair with the seed treatment.
This is another seed treatment that we tested in our on-farm trials, and I want to thank Syngenta for providing this product to us, the Saltro. It's a new seed treatment that's designed targeting sudden death syndrome. And we had 2 trials looking at this. We actually had more, but they didn't go to harvest. And that's a long story, but we will try to get more trials next year. But 2 treatments, we just compared base seed treatment with and without Saltro. And the reason that's important is because Saltro is not being marketed as a standalone product. It should be teamed just like Alevo, teamed with a base seed treatment. So that's why that's our 2 treatment comparison, the base seed treatment with and without Saltro. We did try to pick sites that had SDS symptoms in the past. And what was really unique about our on-farm trials, every time we've tried to do that with Alevo and other products, we have not seen the visible symptoms of SDS, the Foliar symptoms. In this trial or these trials, that was different. We did actually have a really solid infection rate going on and visible toxins in the leaves. So it was kind of interesting this year.
That might just be an important point too, just to mention that SDS is both a root rot and a foliar disease, right?
Yes.
And so we may have root rot occurring without foliar symptoms. And if you have that root rot, you're going to see some yield reduction. It's a little bit like soybean cyst nematode. You might not really know you have the problem, right? If there's no foliar symptoms. So that it can be a little bit tricky in that sense.
And I think, Marty, that's a really good point, because I think that's what we've seen in our Alevo trials is we did not see the foliar symptoms, yet we saw some nice increases in some of those trials. And I think you and I talked about it, and it was the root infection is what the hypothesis was. Nope, that makes a lot of sense. So this is a slide that's taken out at one of our trials down in Calhoun County. Ty Bodice took the trial, the slide, and you can see right here is kind of the dividing line between the treatments. We've got the foliar symptoms here, the toxin symptoms showing up here, where they're less severe over here, and that went throughout the field. So very visible. Actually, both of these sites were in the same field. They were either side of that pivot. The reason we treated them as 2 different trials is right here. We had 2 different varieties, but also the topography was different. On the south side of the pivot, it was more relief, more slope. On the north side of the pivot, you had a more lower topography and flatter topography. So 2 different varieties. They both had good tolerance. This one had Calhoun 2, kind of pay attention to this though. Calhoun 2 did have very good, a little higher rating from the same seed company as the other one did. The other point that I want to make about this background information is we did test for SCN, soybean cyst nematodes, and we found 0 in both sites. And Marty, I was kind of hoping that you would talk about that a little bit, that, we still had SDS pressure, but we didn't have any SCN.
Yeah, so it's certainly not necessary to have soybean cyst nematode to get SDS, right? The SDS fungus is more than capable of causing yield loss and soybean sudden death syndrome on its own. We very often do greenhouse or inoculated field trials where we just have the SDS fungus and no nematode pressure, and we get plenty of SDS disease development. But yeah, if you have the soybean cyst nematode on top of SDS, it tends to exacerbate the soybean sudden death syndrome. And I mean, you're going to lose some yield to nematodes as well. So, it's not a good situation, right, when you're dealing with both issues in the field.
The other 2 things I want to bring up on this is that this was just one pass of a vertical tillage tool and the spring, and I thought I detected some compaction in the sites. So, and compaction and early planting. The other thing about this site is it was planted early. So we had early planting, we had maybe possible compaction, we had a history of SDS. It was kind of like a perfect storm. This is the yield data from the site. And again, this is the 0. This would be the naked seed down here. The height of the bar is the advantage. So we had 4.1 bushel advantage here at Calhoun 2, which is interesting because this was actually the more tolerant variety, but yet we got a little bit bigger response. This was still a tolerant variety, and we still got a nice response. And you average them together, 3.8 bushel advantage to using the product. Very profitable practice in this location. So if you have a history, I think, of severe SDS in the past, I think this is a case where the seed treatments can be very effective. if you've had a severe history of SDS in the past. Marty, would you agree with that, or is there some other?
Yeah, absolutely, Mike. So we've been looking at this for a few years now on the Ilevo seed treatment side of things. In some years where we've put that treatment out in areas where we haven't had SDS, we've seen maybe a 2 bushel response from that. But yes, I would generally agree that in locations with heavy SDS pressure, we typically see more like a 4 bushel response, 3 to 4 bushels. It sort of varies a little bit, right? But yeah, it's, again, in terms of return on investment, you're treating a specific problem with a specific product, right? And so that increases your chance of that return. So addressing that SDS with a specific seed treatment is more likely to result in a positive return.
Good, good. It did affect sudden death syndrome incidents too. We did test this. Ty Bodice went out and took some counts in the field, and incidents, it cut it in half. The seed treatment did. When we added the Saltro, we cut it down to 16%. And it also reduced the disease severity index by half. So yield was affected, and also the actual disease pressure was affected. That photo kind of showed that as well. So we want to get more locations in 2021.
Yeah, so this is, I've got a couple of slides here, 3 slides, I think. Just looking at SDS management across the region. So we collaborate with other states, typically through the North Central Soybean Research Program or United Soybean Board. And so for this particular trial, what I'm trying to do here is develop a risk prediction tool. So we'll take a soil sample and tell you whether you have SDS risk or not based on some DNA technologies we have. And then you can use that information to determine if you need a seed treatment or not. So essentially we had trials conducted primarily across the North Central region, but also as far south as Arkansas and Missouri by the looks of it. We had 58 different locations, differing levels of SDS and soybean cyst nematode pressure. Every trial had a susceptible, what we would consider more susceptible and more resistant variety. We had 3 seed treatments, a base seed treatment, so just a standard fungicide, insecticide treatment, and then that treatment plus Ilevo or plus Saltro. So we're trying to get a sort of determine how effective those SDS treatments are, and then do some comparison between Ilevo and Saltro too. So here's some really recent data from that. On the left here, what we're looking at is in blue, the Folia disease score. So you can see the Folia disease score is higher in that base seed treatment as opposed to Ilevo or the Saltro treatment that significantly reduced the amount of disease in both of those situations. And like we were talking about, this is also a root rot disease, right? So you look at that base seed treatment in gray there, that gray bar is the amount of root rot. So we had more than 20 units of root rot here in the base seed treatment across all these 58 locations compared to Ilevo or Saltro. And again, they're very comparable, very sorts of similar reductions in the amount of root rot. So that's really good to know. And yield response was pretty comparable between Ilevo and Saltro when we looked at all of those locations combined. So yield there is in the orange. So our base seed treatment, we're yielding about 61 bushels on average, and we're seeing up to 62.5 bushels on average there. So about 1.5 bushels or so. So again, this is across all locations. Some of these locations do not have very much SDS pressure, and so we might not see a very significant yield response because of that. So this is just pulling a few locations, and we're doing more analysis on this at the moment. So this is just looking at locations that had a high amount of disease severity. So a score of Folia disease greater than 10 on our scoring system. Again, you can see that reduction in both root rot, well, Folia disease in blue there between the base and the treatments, right? They're the blue bars. And in the root rot, the same thing, right? A reduction. And all of these scores are much more elevated compared to the previous slide set because these are in locations with heavy SDS pressure, right? When we looked at the yield responses, we saw a greater separation of yield response. Now our untreated or our base treatment was down to 58.5 bushels. Our Ilevo is 62.5 and the Saltro is about 61. We're still trying to figure out why we saw slight different responses without Saltro. We know in other locations with heavy SDS pressure, sometimes we've seen Saltro outperform Elevo. So this is early data. What we think might be happening is perhaps that Elevo was providing a little bit better suppression of nematodes compared to the Saltro. That's still somewhat speculative. We're reanalyzing data. We've got a lot of locations and data sets to pull together, and we're only just sort of cranking through that at this point in time. But I think the bottom line, you know, if we're trying to manage any disease, we really should be trying to use the most resistant variety available. The same is true with soybean sudden death syndrome. I would recommend either Elevo or Saltro as a seed treatment. And again, it's certainly more profitable, like we just talked about, in locations with SDS pressure. If you don't have SDS, the chance of return on investment certainly does drop. And always test and manage for soybean cyst nematode, because that can exacerbate problems. And just in terms of bigger picture, trying to manage SDS, it's definitely a very tricky one. It survives very well on corn residue. What we have seen from some work is that extended rotations, you know, 3-4 years, breaking that cropping cycle up with other things, other crops, may potentially reduce the amount of SDS pressure. So I think the question is now, can we use a cover crop? Maybe we can't go away from our soy corn rotation, but can we include a cover crop to increase diversity in the soil and reduce the amount of SDS pressure?
Marty, on that cover crop, I think there was some work from Iowa State that talked about a rotation effect if they included a legume in the rotation, either alfalfa or clover. I can't remember what it was, but it had a legume in the rotation.
Yeah.
Would that favor then, when we're selecting cover crops, should we be trying to select a legume cover crop?
Yeah, so that's a good question. So we would expect that the SDS fungus is probably, in terms of the things it can infect, it might be a little bit more likely to infect some legumes because they may be a little bit more closely related to soybeans. So there's still potential for infection of alfalfa and red clover and other things that were used in that trial. But I think what is happening is that we have a diversity of crops in that system, and we probably are improving soil health, improving the diversity of microbes, and they may either be providing some sort of competition for the SDS fungus or perhaps even feeding on the SDS fungus. So there's some questions we don't know the answer to, and there's no specific cover crop we can recommend at this point in time. Okay. The Iowa State study was sort of, you know, not what we can go out and recommend that, hey, you guys all need to do, you know, 3-4 year rotations between soybean. It's just not necessarily feasible at this point in time. So the question that we need to address now is, can we use some form of cover crop? And that's where we're at in terms of answering that question.
Good, thank you.
Yeah.
So is there any discussion? This is just a close-up of the foliar symptoms of SDS. It is very easy to confuse with brown stem rot, but you can just split the stem and very easily distinguish between the two. But that's the foliar toxins that we were talking about that were present in this Calhoun site that were absent in all of our other trials. Any questions or any discussion at this time? This is a trial, again, it's a product from Syngenta, and we're very thankful to them because they donated a lot of product. You can see the number of sites here. This is Miravis Neo. It's a foliar fungicide, relatively new. We had 14 locations in 2020, and we will conduct this trial again in 2021. We applied the product at 13.7 ounces per acre at the R3 growth stage and at 14 sites. And then we just compared it to an untreated control. So with and without Miravis Neo. This is kind of a disease that Miravis Neo is rated very highly on. It's frog eye leaf spot. And I actually got this picture from one of Marty's trials on campus where they inoculated for frog eye. But Marty, how big of a problem is frog eye leaf spot now? And maybe what's happening, do you think, in the future?
Yeah, it certainly seems that the potential risk across the US here of frog eye may be increasing. So there's a couple of things that are happening. The last few years, we've seen perhaps a little bit more incursion of frog eye leaf spot further north. So I sent some fields from South Dakota, these pictures of fields from South Dakota this year, and they were really, you know, heavily lit up with frog eye leaf spot. And same in Wisconsin, my colleague over there is noticing it moving a little bit further north. You know, typically in Michigan, I personally haven't really seen it at levels where we would typically make a recommended fungicide application for it, but that may be changing. You know, perhaps that's due to changing weather factors. We're not too sure. The other thing that's really important with this disease is that we know that we're seeing resistance to the strobilurin class of chemistry. We've recorded a little bit of that in Michigan, but in other states to our south, that's pretty much everything's resistant to that type of chemistry. The Miravis Neo you're talking about has multiple modes of action in there, so we're not, you know, that still is effective. And most products you use will be mixed mode of action, but that's something we're concerned about too. So it's something we want to keep an eye on. This particular field, like Mike said, we actually grew it in the lab and put it out in this field. It's naturally occurring, so not really changing any dynamics in Michigan, but in this field, we just sort of increase the pressure to look at some different fungicide foliar sprays for management of this. And they certainly do a good job at reducing frog eye leaf spot if you need to spray.
Good. Thank you, Marty. So this is some background data from our 14 locations. You got the locations here, and then you've got the application dates, the spray volume. I kind of want to draw your attention to the spray volume because coverage is a really big issue with foliar products. And the first thing we recommend is lots of gallonage. And usually most labels will say a minimum of 15 gallons per acre. But as you look at these sites and you see we've got the majority of them were applied in lots of water, 20 gallons, some up to 25 or 30. So I think the growers did a really nice job of applying the product in these sites. The other thing that makes me think they did a really good job is when I look at ground speed. There's some work out of an engineer down in Ohio State, Erdl Ozkan, and Erdl found that that we really do want to maintain our ground speed when we're spraying large canopies of soybeans at 10 miles an hour or less. And I know that's going to be hard for some of you to swallow, but that will really improve your coverage, improve your efficacy of your products. So 10 miles an hour or less. And if you look at these columns, this column, you see that we achieved that on most of those trials. So I really am very confident that the trials were conducted well and that. So here's the data from our sites. And we did have a really nice big home run down here in southern Michigan. And actually look at these three. These are all in the southern tier of counties, southwest Michigan, far southwest Michigan. And we had 3 of our 4 statistically significant, or 5, I guess, statistically significant yield increases came from the southern part of the state. And actually, this was down south too, Van Buren. So this other one up in Sanilac County had a response to it, but it wasn't economic. So again, that black bar is the break-even yield. And we had 2 profitable sites. And when we averaged all of the sites together, we end up with 2 bushels per acre, statistically significant, but not profitable. So that's kind of the challenge. And what we put in, just so you know, we did charge the full commercial application rate here. So those of you that apply the product yourself, you can probably reduce that a little bit. We did charge the full $8 an acre. But still, even if you're applying it yourself, you do need to put a number in there, probably more like $5 an acre for your application cost. And then we also did include a $20 an acre cost for the product. So that's how we arrived at that economic break-even point.
And Mike, I just comment too from our experience with foliar fungicides. So just basically going out there and spraying with no real disease pressure, right? We're not trying to control white mold. We're just in a location with no real detectable disease pressure. We've seen 8-10 bushel increases at times with a foliar fungicide, but we've certainly seen lots of negative data too, or instances where we just do not really see much of a response, maybe 1 or 2 bushels. So again, it seems to vary on location. There might be other factors there that are hard to measure. And certainly, this goes to our action point again. If you're treating a disease, if you actually need to manage frog eye leaf spot, yeah, you're far more likely to see a return on investment as opposed to just spraying because you have time to spray or you just sort of want to spray. I'm not saying don't, I'm saying think about what you're doing. And I think it's really important to include some check strips. If you are making the switch to, you know, spraying half of your farm or a particular field, try and leave some check strips in there to get a sense of what is your return on that investment. I know that can be difficult at times, but I think it really does, you know, has incredible value, you know, to knowing what was my return on that investment. There are some tricks with that, right? You've got to be a little bit careful about where you leave those check strips and whatnot, you know, field edges versus in the middle, da, da, da. But I think some form of check is pretty helpful just to get a sense of, is this working for me or not?
I agree, Marty, and that to me is one of the strengths of our replicated strip trials with the on-farm program. If somebody's interested in finding out how Mirivus Neo performs on their farm in 2021, boy, just contact me and we'll do a replicated trial, and you will know within 90% certainty did the product how the product performed on your farm and under your conditions. And I agree with Marty 100%. This is very site specific. We have one of these sites up in Sanilac County that actually responded to a different fungicide 3 years in a row or 2 years in a row, and then responded favorably to the Mirivus Neo. So it's not so much the product as it was the site. And so we do need to try to find out what's going on with that producer. So this is our white mold foliar fungicide comparison trial. And actually there's a word that's missing there. It's really a program, a fungicide program comparison trial. And I'll show you what we did. We did it for 2 years, but basically what we did is there's a treatment that's being recommended up in Huron County. Prominent agronomist up there has been recommending the use of Cobra followed by Approach, and had very good luck up in that part of the world, which is a hotbed for white mold. We have had very good luck with Propulse in our strip trials. That seems to be an effective trial in our strip trials treatment. So we compared those two different programs, a single application of Propulse versus this Cobra followed by Approach program. And we compared those to an untreated control. We did this at 3 locations in 19, and then another 4 locations in 2020. Cobra was applied prior to R1. Basically, when you first see a first blossom, that's when we want the Cobra to be put on. And then the fungicides were applied 10 to 14 days after that. And all of the sites had a history of white mold, but due to soil and climate conditions, none of them actually had white mold. So the way to interpret this data is, how did these fungicide programs perform in the absence of white mold? So that's the 3 different mushrooms. This is the correct one. This is actually going to be the white mold apothecia, and you can see they're really, really small. This is a quarter of an inch. Quarter of an inch would be that biggest one. That would be about a quarter of an inch, so they can be really tiny. And I always thought they were described as a golf tee, so I was looking for something bigger and something taller, but they're going to be really close to the ground, really close to the soil surface. And I guess all of these are. This is the one that disturbs me. The bird's nest, I've been out with some prominent producers in the last couple of years that have battled white mold, that are familiar with the disease and are good managers at managing it. And they have mistakenly identified this mature bird's nest fungus as the white mold fungus, so or mushroom. So the way you distinguish these two is, I don't know if you can see it from this picture, but in the bird's nest, you're going to actually see little eggs, little black eggs inside there. And that is just a really clear distinguishing factor. So that is not the white mold mushroom. And Marty, to my knowledge, it doesn't cause any diseases that are of any concern in soybeans.
That's right. If we didn't have fungi in the world, we'd be drowning in organic matter. So fungi are really, really important for the breakdown of corn residue, soybean residue, assimilation of nutrients. There's a whole host of positive things that fungi and bacteria do for us. It's only a very few of them that cause problems. And so these bird's nest fungi are completely harmless. There's no negative impact, probably a very positive one. And so, we really want to make sure we're out there trying to identify things that we know what we're looking for. And I get it. The white mold fungus is hard. It is hard to find those mushrooms out in the field.
It really is. I got to tell you, because I actually took this photo, but I never would have found them without Marty's help. I was traveling with Marty, and we found a field in Ionia County, and I took some pictures. Marty provided the other 2 pictures to me, and I really appreciate that. This is just a really devastated field up in Sanilac County in 2014, border to border white mold, probably 95, 99% incidence, just terrible. Marty, this is an important concept. If you would discuss this with us.
Yeah, so we do a lot of white mold trials. We screen a lot of different fungicide products, timing, which we won't get into all of that again today. But the other thing that we do, and have done more of that recently, is screening varieties. So we work with the MSU soybean breeder as well, and help him have an environment that he can screen his varieties to develop new lines. But we've also done some work for commercial companies. And so this is one of those companies, and it just shows you a spread of susceptibility in their germplasm or in their different soybean varieties that they have. So along the x-axis here, the bottom axis, you can see the amount of white mold disease. So we're running from 0, very, very low pressure, up to 50. So 50 is a plot that looks very, very bad. A lot of disease in that particular plot. And so you can see here there's a very strong correlation between the amount of white mold and the yield. So as Mike draws out, follows that line, the amount of disease is increasing, and so the yield is decreasing pretty rapidly. I labeled these as well with the maturity group, just so you had that information. We can find mid-2s that are very susceptible. We can find mid-2s that are very resistant to disease. And we can find early maturity groups that are very susceptible as well. Not in this particular instance, perhaps. I mean, there's one it's in the middle there, a 1.8, that's about 25 on this scale. So that was pretty heavily diseased. So I wouldn't necessarily be trying to pick a maturity group to avoid white mold. But I would really, again, be talking with your seed dealer, making sure you're getting the best information on the best available resistant varieties that are there. And there's no variety that's completely resistant to disease, right? All of these best performing varieties up in the top left of that picture there still had some disease, but they yielded really well. So it really wasn't much of a problem for them. And this is an irrigated location at Montcalm. We use irrigation because that ensures that we have a successful screening environment every year, right? But weather is a very, very strong factor as to whether we're going to see white mold develop or not. And we're going to talk about that in a minute here too.
Good. Well, this is just the overwintering structure, the sclerotia, that can last for 5 to 8 years in the soil if we bury it any deeper than 2 inches. So that's the culprit that just survives from year to year. This is the background information that we collected from the trials. And you can see we tried to, like Marty says, managing diseases with resistance or tolerance selection is really one of our best lines of defense. So we want to give that information to you. We've got the application dates over here for all the trials. The one thing I would like to point out is that you have to be very careful in using some of these scales because in some instances, you've got like right here, 1 on this scale is poor and 9 is excellent, but you go down to here, this company, and 1 is excellent and 9 is poor. So you have to be really careful when you're looking at these catalogs. And I think I would also caution you to not try to extrapolate across companies. I think the ratings are very good within a company's line, but if you try to compare a Pioneer variety to an Asgrow variety, it might be more difficult to do that. The ratings may not be the same. So this is our data and from all the locations. And again, please keep in mind that there was no white mold pressure at any of the sites. In 2019, we did see some really nice responses at 2 locations here in Sanilac and in Allegan County to the Pro Pulse. It really was profitable at both locations. The Cobra followed by Approach was not. down here, this is really an anomaly. I don't know what happened, but we did actually lose 8 bushels per acre here at this Van Buren site in 2020. When I look at the phenology model of when the plants were planted and the maturity group for that bean, they were well past the stage that it should have been applied. They were almost to R3. They were certainly into R2 towards the end of R2 and almost to R3 by the time this was applied to Cobra. So I think there was significant damage done by the Cobra at that location, is my hypothesis there.
Mike, if I could jump in really quick too.
Yes.
So, you know, the fungicide timing has got to really be between R1 and R3, right? Because we're trying to control infection of the flowers. That's really the important piece. So I completely agree with you. And that's interesting. You figured out that timing issue there. So putting on that Cobra at R1 or a little bit before, the idea is to prime the plant for resistance to disease during that flowering period. So if the application was too late, you've missed that primary infection period, and there's going to be potentially maybe more crop injury. I guess I don't know enough about that particular situation, but anyway, yeah, that's a very good point, Mike.
The other point I want to make here is that Marty, actually in his small plot work up in Montcalm, he inoculates for, or doesn't have to inoculate, but he has lots of white mold pressure, and he does irrigate to foster the white mold development. In a situation where white mold pressure is heavy, Cobra has given you nice results, Marty, in your small plot research.
Oh, it's been amazing. It's been as good as the best fungicides. So again, though, that's in situations where the white mold pressure is incredibly heavy.
Yep.
We see a very positive response to that Cobra treatment. So my caveat is, yes, if you've got very low white mold pressure, or you're not expecting that much this year, I'd be a lot more careful with it. You know, but if you know you've got a problem field, we're in a really wet season, you know you're going to have an issue, that's when I think it might be worth trying. And again, leaving check strips, right?
Yep. Nope, that sounds good. This will probably be our last year of this trial, but we will do something with white mold in 2021. We just haven't decided that yet. We wait to get feedback from producers before we make that decision. Here's some action points, Marty.
Yeah, so again, like we sort of hammered that message home with that slide, right? Showing you that it's really important to select the most resistant varieties you can for white mold management. Apply fungicides at the correct timing, and we hope that sporecaster model will help you refine that. In our trials, sometimes we'll see that an R1 application is better than an R3 application. Right? Again, it depends on when those spores are being released and the weather environments. And so that's why we believe that sporecaster model is important, and does, you know, deserve some attention to make sure people are using it, or at least aware of it, and certainly improving it for irrigated situations. Select those fungicides that are effective. I mentioned the cropprotectionnetwork.org. So just Google crop protection network. And you should be able to find that website and navigate to the most up-to-date soybean foliar fungicide efficacy chart. I guess the only other one, Lectivar, is a similar product to Omega. It's got the similar active ingredients, but I think we mentioned all those that would rank good or above. There certainly are other products that would perhaps rank as fair, and they may be cheaper. So there's another tool out there called Spore Buster, another smartphone app that you can download to play with the economics. That's a little bit newer in the development, but that might be something you may be interested in doing. And we want to mention too that Sporecaster app is very easy to download if you have a smartphone. Just go to your Google Play or your Apple Store and type in Sporecaster, and you'll be able to find it and download it for free. And then Mike's done some really cool work on reduced planting rates, and we've been doing that work as well at Montcalm, just demonstrating the importance of reducing planting rates. We've also looked at row spacing. That can make a huge difference. I realized that, there's other factors to that. You can't just rush out and buy a different planter, and 30-inch rows brings some potential issues, perhaps with weed control. So there's other factors to consider, but certainly, 30-inch tends to reduce white mold development. Again, weather is the driving factor here. If you have very unfavorable weather or favorable for white mold, it doesn't matter what you do, you're going to get some level of white mold, right? And so, and then we had a comment there about irrigation. That certainly increases the risk, right? Of course. A field history, I guess we didn't put that down, Mike, when we were chatting. So, knowing what your history of white mold is, if you've never ever had white mold before, it's unlikely you're going to have a significant issue. Maybe 2014, again, was a bit of an exception to that. We had such terrible environmental conditions up in the thumb. We had producers that had never grown soybean in a field before, had soybeans and got a lot of white mold. I think because it was so overcast and so wet during July and August, we had spore movement from field to field, and we don't typically think about that as a problem with white mold. And then what would you like to say about tillage and cover crops, Mike? I'll let you get in there.
Just that I have had anecdotal data from producers that are long-term no-till producers that just have not had white mold. They will grow the most susceptible varieties and do not have white mold when all of their neighbors do. Same soil type, same everything, same rotation. It's just the tillage, I think, has several methods for maybe there's a hypothesis. One of them is if you think of the sclerotia as weed seed, that they will only germinate in that top 2 inches of soil. So, and that they have to be in that top 2 inches or on the surface to germinate. Well, if one of the best strategies, I think, that I was recommending really hard after 2014 is, okay, we generated an unprecedented amount of sclerotia. Let's just leave those on the surface if we can. Leave them to where they would environmentally degrade or germinate or get predated or whatever. Just leave them on the surface and then try to no-till a small grain into that fall. And boy, that would just encourage them to germinate. You'd burn that crop out. You wouldn't be burying them into the soil. So I think tillage is a mixed bag. The rule of thumb I always heard with tillage and white mold is if you're going to be planting a susceptible crop after a bad white mold year, then you till deep because you want to bury that year's crop of sclerotia before next year's susceptible crop. If you're going to be planting a resistant variety or a crop that's not susceptible to white mold, boy, no-till them, leave them right on the surface and let them degrade or germinate and try to infest that non-susceptible crop. Cover crops, there's a little bit of work done out on, well, there's some work that shows that cereal rye can have some effect, but it's not, we need to look at it more, Marty. I think it was actually from Wisconsin. It was really interesting with small grains planted as a cover crop and allowed to grow in the spring. What they did is they actually promoted the apothecia production earlier than when the soybean crop was susceptible. So the blossoms were not out on the soybean crop yet, but yet the mushrooms were forming and releasing their spores. So it was a timing incompatibility that was really fostered by the cover crop use.
Yeah, I think that was actually from one of my former advisors out of Cornell. But that doesn't really matter. Wisconsin may have looked at that too. But yeah, there appears to be some really interesting possibilities there as well with those cover crops. Yeah.
So, and primarily small grain cover crops, something that's post-seeded, either wheat or cereal rye, something like that. No, that's all I had on that.
Okay.
I do want to mention that our report has been mailed. I just got an email while we were talking that somebody got their report, thought it looked pretty good, and so that is available. But if you haven't received it yet, if you're from agribusiness, now you will not get it mailed to you. It's only mailed to the soybean producers through the checkoff because they pay into the checkoff. But if you want one, you can probably request it. But also, if you just go online, you can print it. It prints really nicely from the site. So that would be this year's on-farm research report. And I do want to thank Marty for his collaboration on many of the trials. He's given us a lot of feedback, a lot of input, and helped us with our design. So thank you, Marty.
Oh, likewise, it's really helpful to have translation, or if we find something in small plots, to chat it over and try and get it out onto on-farm trials just for demonstration and testing purposes at that scale. I think that's really important, and we need to keep doing that. And just a quick shout out, thank you to the Michigan Soybean Committee. Thanks for the support, because without that, we're not able to do what we do.
Now, I'm only going to focus on the 8 projects that are highlighted or bolded here. And the other ones are more disease management trials that will be highlighted in this other presentation. First one I want to talk about is our planting system comparison. This is an interesting trial, and its producers really drove this. They wanted to see us look at different row spacings, but not only different row spacings, but different pieces of planting equipment. how does my air seeder compare to my planter, for example? Or how does my box drill compare to my air seeder? Or just various combinations. So of row spacings, of delivery systems, and ground working parts. So that's why we call it a planting system comparison and not simply a row spacing comparison. Here's what we did in 2020. We only had one location, but we compared a 30-inch row corn planter to a 15-inch row John Deere air seeder. And one location, but it was 8 replications. Very reliable data, replicated 8 times across the field. And we did keep the planting rates, at least we tried to keep the planting rates the same between the 2 treatments. And we did take final stand counts at the end of the year. So this is some background information. You're going to see a very similar table for all of my trials that I present today. And what we do is we try to capture the background information that might help us explain why or why not a difference occurred between the treatments. And what this slide shows is the actual data. So here's how a lot of the slides will be presented. The locations will be here, and then the treatments will be the next 2 columns. And so here we've got the John Deere planter, and here we've got the air seeder. You can see the yields are essentially the same. You know, not broad enough to be a statistically significant difference, but they're essentially the same. So if they're essentially the same and we apply the gross income to it, they're going to be roughly the same as well. Now, the one thing I do want to point out, though, is that there is not a difference in the purchase costs of the 2 pieces of equipment we evaluated, but there is probably a higher operating cost to the air seeder. It is going to have more draft requirements, so it's going to need 25% more horsepower. And it also is probably going to have more ground working parts to replace. So we would expect, we did capture final plant stands, as I mentioned. And if you look at this here, this first column being the planter, it did have a statistically higher plant stand at harvest than what the air seeder did. And we would expect that. It's got a better metering system. It has better opening system, better closing system. We would really expect to see better seed to soil contact and better depth placement with that planter than we would with the air seeder. And consequently, that's what we saw. Now, it wasn't a big difference, 2,500 seeds per acre, or plants per acre, but it was a difference. This is our strongest database. We've been doing this trial. It's our planting rate trial. We've been doing this for 6 years. And you can see we're pretty heavy up in the thumb, but we are getting better geographic distribution elsewhere in the state in the later years of the trial. We actually had 58 locations from 2015 to 2020. Producers across the state really identified this as the highest priority project. And the reason they felt that way is because income was down, commodity prices were down, and seed is a very high variable cost for producers. So if we can reduce that seed cost, that might help and positively affect income. The other thing that was driving this was white mold. 2014 was the worst white mold year we've ever seen in Michigan, and there is data that supports the hypothesis that reducing our planting rates will help us manage white mold in a very bad white mold situation. So here's our 4 planting rates. They wanted to see how low we could go. We went down to 80,000 all the way up to 160,000. We put these into different environments with different pieces of planting equipment. Here's a no-till planter, actually a split row planter, planting into no-till conditions. It is planting in 30-inch rows. Here's another trial. This is one that was planted into some worked ground with a 22-inch row planter. And here's an air seeder planting into no-till conditions. So different pieces of planting equipment, different tillage conditions. This is our yield data. And what this is it shows the individual years. The bars are the individual years within. So this is the 80,000 planting rate for all 6 years. And then the darker is the newest year, and the lightest bar represents 2015. So going from dark to light would be going from most current to the oldest trials. And that's the same for each one of the planting rates. The biggest thing that this shows is the year-to-year variability that we saw. This is why we want to conduct our trials over multiple years. Look at that, the yield variability just within that one planting rate, how much the yields varied. So what the lines are then, the lines represent the average income for each one of those years. And there's one trend that is very, very obvious when you look at this. If you look over here at the 160,000 planting rate, the yield, the income really tails off consistently every year in the 160,000 planting rate. It was never the most profitable site for a given year. Now you look at 130, and it was actually the most profitable in 2020. But for all the most years, it was the lower two seeding rates that were actually the most profitable. So that's kind of interesting data there. Now what this slide is this is my attempt to take our management practices over here in the left-hand column and see how they interacted with the planting rates. So the first two are going to be row spacing. And then what this is this is the percentage of trials where the highest planting rate beat the lowest planting rate. So if we just look at this first one where we're looking at row spacing, we have 56% of the time in 15-inch rows did the higher planting rate beat the lower planting rate. If we widen the rows out, it's only 21% of the time that the higher planting rate beat the lower planting rate. What about planting equipment? So if we're planting with the drill, those trials that were planted with the drill, 50% of the time, the 160,000 planting rate beat the 80,000. If we planted with better equipment, more of a precision equipment, the planter, that drops down to only 33% of the time. What about no-till? If we're planting in no-till conditions, 50% of the time, we saw an advantage to the higher planting rate. When we drop down to a tilled condition, that dropped down to only 21% of the time. Seed treatment is kind of an anomaly. The only thing I would tell you is that most of the vast majority, 90% plus of our trials were planted with a base seed treatment. And so that one was kind of hard to sort out. So I don't know if that means anything, but this one is kind of consistent with what we would expect, is if we're planting after June 1st, there was 67% of the trials did the higher planting rate win. And if we planted before June 1st, only 43% of the time did the higher planting rate win. So very consistent there. This is our next trial I want to talk about. This is one that there's a lot of interest in. It's our planting date trial. And there's a lot of interest in this on both ends of the spectrum. There's some growers in southern Michigan that want to plant ultra early. And when I say ultra early, I mean before April 15th. And then there's other growers in the state that are hesitant to plant before May 1st. And so we just want to get some data out there that shows what the benefits are and maybe what the risks are of early planting. So to do this, we had early planting dates were compared to normal planting dates for the area at 3 sites in 2019 and another 8 locations in 2020. The early planting dates in the Branch County locations are considered ultra early. They were prior to April 15th. The early planting dates at all the other locations are very consistent with MSU's current recommendations of planting your soybeans if the conditions are suitable during that last week of April. So we also want to thank Dr. Manny Singh for providing some funds to conduct these trials. And Manny was able to secure those funds from a grant from the North Central Soybean Research Project. Dr. Singh, thank you very much. I want to put this slide up because this shows one of the perceived risks of planting early, is what happens if my beans make it out of the ground and emerge, and then I get a killing frost or freeze event. And that's exactly what happened here in 2020. This plant is not going to make it because you can tell by the discolored cotyledons, but the worst damage is right here below the cotyledons, right here in the hypocotyl. This is what's really signifying death. All the meristematic tissue is above that. So if you get freeze injury there, that plant is toast and is not going to make it. This is an example of a plant from the same field where the shoot was, the primary shoot was killed or is dying, and it was stressed to the point that now we're getting new growth out of this axillary nodes here. And we're getting new growth out of these axillary buds that are here at this node. And that's a really good thing. This plant is going to produce a viable plant. And so this is one of the reasons why you don't give up on a stand right after a freeze event. You do need to go out probably as much as a week after the freeze event and see if you see this type of growth happening. If you do, that's a viable plant that will produce a viable yield. This is the background information that we captured from the trial. And you can see that we've captured everything that we think might affect the outcome. So we've got the planting dates for both the early planting date and the normal planting date, tillage, texture, planting equipment, seed treatment, and row spacing. When we look at the yield data, this is a column represents the early planting date. This is the normal planting date. This would be the branch site all the way across. And you can see the first 3 sites did have a statistically significant yield increase to early planting. The rest of the sites, it was pretty flat. There really was no advantage or disadvantage to planting early, statistically significant advantage. But some of those numerical increases or bushel increases can be quite big. Here in Branch County, we had a 6.5 bushel increase. And the lowest increase that we had actually was statistically significant, was almost 4 bushels. So it can be a big deal. When we average them all together, it's about a 2 bushel per acre increase. And when we put economics onto that, we gained about 20 bucks an acre by planting early. 20 bucks an acre by planting early. So that's a big deal. We can't always count on that. It's there sometimes, like here, 3 out of these trials it was, the others it wasn't. But one thing I want to point your direction to is that there was no statistically significant yield losses. So we had upside potential, but no downside potential in these trials. And I think that's worth noting. This is just a graphical representation of that same data. This line is representing the normal planting date. The bar is then the yield increase that we would have gotten, or the yield difference, by planting early. So here's the home runs right here at the beginning. These are kind of maybe just noise. They weren't statistically significant. When we average them all together, though, we end up with a 2 bushel advantage. So that's very real. And again, no significant risk. A little bit of noise, but it was not statistically significant. So early planting can be a really important practice for many growers in Michigan. I actually view it as a risk management practice. Instead of being highly risky, I look at it as a risk management practice. And the reason I say that is because sometimes our best planting conditions are early, and we get into some wet conditions in the month of May. So I think it's important that we not let those really good early planting conditions get away from us, and try to get our seed planted in there if we can. Basically, we're extending our planting window by planting early. This is our row spacing comparison, and it's a really interesting trial because a lot of data that was done prior to our work in row spacings actually commingled different types of planting equipment, where our trial is truly just a row spacing comparison. We're taking the exact same piece of equipment and just changing the row spacing on that piece of equipment. And where much of the other work was done with drills versus planters and those kind of things, what we would call a planting system comparison. The reason this is important is because we're comparing 2 of the most common row spacings in the state of Michigan, 15-inch rows versus 30-inch rows. And the reason that's important is growers are trying to make some capital purchase decisions on their next planter. Do they go with an inner plant or a split row planter and give up some width for the same amount of money, or do they plant in 30-inch rows and get a wider planter for the same amount of money? So those are the decisions that need to be made, and hopefully our data will help producers make that decision. This is a typical piece of planting equipment. It's a John Deere planter with splitter units. The splitter units are raised, and so at this point in time, he's planting in 30-inch rows. So we did compare the two row spacings, 15 inches versus 30 inches, at 2 locations in 2019, and then another 6 locations in 2020. All trials were planted with the exact same piece of planter in both treatments. All we did is raise or lower the splitter or interplant units. Our goal was to keep the planting rates the same between the 2 row spacings, and we achieved that for the most part. This is the background information for the trials. And the ones I really want to draw your attention to is, look, mostly of the planters were John Deere, all of them were John Deere planters. And then the other thing I want to draw your attention to is the planting dates. These are really good planting dates for this trial, because if you plant much later than that, you start to bias in favor of the narrower rows. So I think we're good there. And then the last thing I wanted to draw your attention to is the varieties and the plant type. Typically, if we're planting in wider rows, we want to have a variety that has the capability of branching out and filling in those inter-row spaces. And almost all the varieties that we plant in the state of Michigan have that capability. A medium bush type is probably our most common variety sold in the state of Michigan. This is the actual yield data. We have our locations in this first column. Here's the 15-inch row configuration, and there's the 30-inch row configuration. So we can see that there's 3 trials where we had statistically significant yield increases because of the 15-inch rows. And I think that's really, really interesting. And some of the numbers come pretty large. This one up in Shiawassee County was 5 bushels per acre advantage. And the lowest we got was just under 2 bushel per acre advantage of the statistically significant yield increases. When we average them all together, we end up with a 2.1 bushel advantage to the narrower rows. Now, the economics on this are a little bit challenging, a little bit different. It actually turned out to be a partial budget analysis, and Roger Betts, farm management educator with MSU Extension, helped me do this partial budget. Roger and I decided that we were going to use, for a market price, $9 per bushel instead of the $10.40 that we used for everything else. And the reason is, this is a long range capital purchase decision. And so we wanted to use the long range projection numbers, which unfortunately are right around that $9 per bushel, at least at the time we did this analysis. So if we use the assumptions of a 12-24 interplant planter, given 500 acres of soybeans for 10 years, it's going to cost you an additional $18 per acre to own the interplant planter versus a 30-inch row planter. An additional $18 per acre every acre you plant. It has to make up for that if you're thinking about an interplant planter. So that's the economics on it. You can see, based on these numbers here, we were able to achieve that 2.1 bushel yield advantage. So there is a slight economic increase to the 15-inch row planter. This is just a graphical representation of that same data. And the way to look at this, again, the 0 is going to be the 30-inch rows, and the bars are going to be the yield increase, yield increase that was earned by going to the 15-inch rows. And we have to have about 2 bushels per acre to break even to cover that $18 per acre additional cost. And when we average all the sites together, we do achieve that. And we achieved it at 2 of the individual sites, but none of the others. So that's the economics on it. I would say that there's some other factors that go into play in this decision. One of the factors to consider is the wide rows. 30-inch rows tend to look better in maybe marginal soil conditions, planting conditions, and they may look better on, actually, no, I'm sorry, they're going to actually look better in higher fertility conditions. When you have very productive soils, the 30-inch rows look pretty good, especially if mold is a problem, then 30-inch rows will be better. If crusting is a problem and emergence is a problem, the 30-inch rows is typically a better spacing because the plants can help themselves out of the ground and crack the crust. The 15-inch rows are typically going to shine when planting becomes delayed. That's when they would shine. Probably after June 1st, I would say, you might start to see more of an advantage to the 15-inch rows. The other thing to think about is the interplant planter versus a 30-inch row corn planter. You can actually buy a 25% wider planter. So you could buy a 16-row 30-inch row planter for the same price that you paid for a 12-24 interplant planter. And so what does that do for you? Well, it may help you with challenging planting conditions. You might be able to increase your planting capacity by 25%. So it is a complicated decision. We're trying to provide the yield data to help growers make that decision. This is the stand counts from our trials. Ty Bodice again took these stand counts. And there is a couple of anomalies here I want to mention. The Saginaw County one is really interesting because what happened is we end up with a half a stand in the 30-inch rows versus the 15-inch rows. And this was just human error. What happened here is when we went from the 15-inch rows to the 30-inch rows, we did not adjust the seeding rate. So we kept the seeding rate static, but we planted half the number of rows, so we end up with half a stand. Now in Tuscola 20, the reason that there's an anomaly there is what happened is in the guidance system wasn't quite working right. And so what happened is some of our 15-inch rows got planted on the old 30-inch corn row, and that led to some emergence and stand problems. So those are the two anomalies. Other than that, I think we achieved our goal of keeping the planting rates about the same between the two treatments. This is our next trial I want to talk about. It's one that we've done for the last 2 years. This is our tillage trial. And well, you can see the locations there. We had 2 locations in 19 and 4 locations in 20. Pretty good geographic distribution. I think we would like to have more of these trials if we could. I think there's a lot of interest in what are the economics of tilling our soil prior to planting soybeans. This is just a picture of kind of a common trend is these high-speed discs. This happens to be the Degelman ProTill, but the high-speed discs are kind of a common thing, a single pass. You can see how much they do work the soil with just a single pass. And then we're able to plant our soybeans right into that conservation tillage system. So when we first started this trial, we were looking at a single spring pass compared to an untilled control. Well, that's not, in reality, that's not what we ended up with in one of our locations. We do have a 4th site in 2020 that did compare chisel plowing followed by a soil finisher versus an untilled control. So most of the trials were a single tillage pass in the spring, with the one exception in Berry County. So this is our sites across here. You've got the years after them and the counties that they're in. You got the tillage tools that were compared. So we had vertical tillage. Here's the site that was chiseled and then fit. We got an older school disc. We've got the new high-speed disc. This was the same grower that participated in 20, also did it in 19. And then in CAS, we just had a single pass of a soil finisher. Here's our planting equipment. In only 2 cases, we used a box drill, and then 1 case we used an air seeder. So all times we followed corn. Good planting dates. We had a couple of late planting dates actually in there. Seed treatments that were listed, and then your row spacing. And again, the reason we cite this information is to try to help us discern why or why not did we have a difference from tillage. So this is the yield data from all of the sites. This Ottawa 20 site was the one that showed us a statistically significant yield increase this year, 3.5 bushels per acre. for just a single pass of a vertical tillage tool. So pretty impressive. And but none of the other sites really gave us anything. We didn't lose any yield by tilling, but we really didn't gain any yield by tilling at the other sites. Even at this berry site that was tilled more intensively, we really didn't gain anything. Numerically, a 1.6 bushel advantage, but it was not statistically significant. We averaged those all together, and we do have a statistically significant yield increase of 1.3 bushels per acre to tillage, and that's because this one was so large up here, and we averaged it all out. So you apply the economics onto that. Just a single tillage pass would cost about $14 an acre. You subtract that from your gross income over here, and you end up with basically flat, no changes in income there. So this is just a graphical representation of it. The thing that this slide does show is the break even across all the locations. And we do just break even, as I talked about earlier, across all of our environments. We had the one profitable site. This site is not profitable because this is the berry site, and we had 2 passes there. We would have had to have a 3 bushel increase to be profitable at that site. So what happened to stands? You know, you think typically tillage is going to improve your stands. Well, it did in one site here in Berry County. It did actually increase our stands quite a bit, 16,000 plants per acre. But then in Isabella, it actually cost us about 3,000 plants per acre. So pretty interesting. I don't know exactly why that is, but they kind of flip-flopped. You average them all together, it's flat as can be. Basically, in our trials, tillage did not have a significant effect on stand. This is the next trial that I'd like to talk about. This is our rye termination timing trial, or another way to think about it is planting green. Rye is one of the most common cover crops that we have in the state and has many beneficial attributes to it. One of them that's being most currently explored is its effect on reducing mare's tail pressure. Dr. Christy Sprague has been looking at this and has seen some very good results. And planting green seems to really make those results even more favorable. So that's one of the reasons we're looking at this. So we had 3 locations and 2 different treatments. We terminated the rye prior to planting, and then we terminated the rye after planting, or planted green. And we took final stand counts as well as yields at these 3 locations. This is a picture that Dean Boss gave me, and an excellent picture of us showing planting one of the trials into the green rye cover crop. This is another picture that Dean provided, and it shows the soybean crop emerging and growing in the treatment that was killed after planting. This is some of the background data that we collected from our 3 sites. We had 2 in Jackson County and then 1 in Sanilac County. And there's our planting dates, our planting rates, the planting equipment, and then the 2 termination dates for each trial. And then we also have the herbicides that were used to burn down the rye. This is the yield data from each site. Here's Jackson, Sanilac, and Jackson 2. And the dark green bar is killed prior to planting. The light green bar is the planting green, or where it was killed, the rye was killed after planting. So you can see at this first site, just flat as can be, we do see a little bit of a numerical trend to where the planting green did not perform as well as planting earlier at 2 sites. We average them all together and it's pretty flat. In none of these sites do we see any statistically significant differences. So what that really means is if they're not statistically different, there really is no difference between the treatments, even though there's some slight numerical values. So yield was really not affected at these sites, which is really a good thing. It gives you a much bigger window to control the rye cover crop at the very least. And if you're battling mare's tail, it actually gives you a long, better window to plant green. So how did it affect stands? How does the timing that we controlled our rye cover crop affect our final stands? Well, as you can see in these 3 locations, it really did not have any effect at all. Very minimal effect. We averaged them all together, and again, no statistically significant differences there. Now, we did lose a couple of our trials this year. One was in Allegan County, and it was lost due to some significant slug pressure that we had in the field. And so the grower went back and interplanted between the rows and to make up for the slug injury. Another situation was lost at the Kellogg Biological Station, because what happened there is you saw that picture of the standing rye and the soybeans coming up through that. Well, at harvest time, the cutter bar had a hard time managing that standing rye, and it would plug up. And when it plugged, we had to stop. And when we stopped, it messed up the yield monitor data. So that's how come we lost that trial. Although the farm manager there, Brooke Wilkie, really shared some really important observations from his trial. The first one was the planting green did help suppress the mare's tail in the trial. He definitely verified that. It wasn't complete control, but it did suppress it. The second thing was that he noted is that there was more armyworm, where he allowed the cover crop to go a little bit longer and planted green. There was more armyworm pressure in those fields, those strips. The third thing he noticed is wherever he planted green, he did have a maturity delay. Same variety was planted of soybeans, but the planting green treatments did cause a maturity delay. So those are some of the observations. Oh, there was one more observation that Brooke made, is he feels like if we could have maybe crimped or rolled that planting green treatment prior to planting, that would have made a difference. And I think that might also improve the mare's tail control, because you're going to have more of a mat down there and provide more shading effect on any weeds that might try to emerge. So those are some of Brooke's observations. This is a trial we want to do in the future, so we are hoping that we can get more locations next year. This is an interesting trial. This is a soil fertility trial, actually a foliar fertilizer trial that we implemented because producers across the state have told us that they are very interested in fertilizing their crop more intensively. And foliar fertilizers is something that are specifically named in our surveys. So this is our attempt to try to meet that need. And so we're looking at a product from Wilbur Ellis. It's called End-Demand 88. It's a complete foliar fertilizer at 10-8-8 with 2% sulfur and some trace micronutrients. And we applied this at 1 quart per acre and tank mixed this directly with our post-emergence herbicides and in that application that we're going to be doing anyways. So it's efficient and it reduces our costs. So that's why we did it this way. And supposedly this product is compatible with every post-emergence herbicide that was on the market in 2020. We had 9 locations across the state, so we feel really good about it. Too fast. This is our background soil test data. We tested all the sites, collected soil samples, and that is a perk that our cooperators get. They get a soil sample from all of the fields that we do. And so you can see when it's bolded, that means that the nutrient level is low or very low. And so for phosphorus, we had actually 3 of our sites here in phosphorus that were low. And then over here in sulfur, we actually had 9 of our sites that were low in soil test sulfur. This is the background information that we collect from all of our trials to try to explain why or why not are we seeing differences. And we've got our planting date, our fertilizer applied, the herbicides that we applied, and also the rates, and then the application dates. This is the actual data, and we had this one site over here on the far left up in Sanilac County that gave us 4.2 bushels per acre consistently across the reps. So 4.2 bushels per acre was statistically significant at this one location. However, it was not statistically significant at any of the other locations, kind of just your typical piano graph type of thing where you'll get some above the bar, some below the bar, and that's what we saw here. So, but when you average them all together, we do get almost a bushel per acre statistically significant advantage, and which is a good thing because when you look at, we only needed 0.4 of a bushel to pay for the product. So we do need to look at this in more in the future, and we will be looking at it in 2021. So really appreciate the support and look forward to seeing more trials of this. This is our Infero biological and humic acid trial. And the reason we conducted this trial is producers across the state, some producers have Infero capability on their planters or drills, and they want to utilize that equipment. They want to find a product or products that will go through there and be applied directly in contact with the seed and make them some money, essentially. So we had 7 locations, good geographic distribution. Again, this is 2 products that were given to us by Wilbur Ellis. This is their answer to an Infero application on soybeans. And there's 2 products that were tank mix compatible here. Nutrio Unlock is a biological product. It's applied at 1 pint per acre. And then Puric Prime Max is a humic acid product applied at 1 pint per acre. These 2 products do have to be applied in furrow and not in a 2x2 placement. If you're looking at 2x2, we may have to find a different product to put in there. Wilbur Ellis was quite explicit that this is not designed for a 2x2 application. This is the yield data from the trial. And you can see over here in the far left, we had 2 sites that gave us statistically significant yield increases. These are on lake bed soils, finer textured soils with more organic matter and more clay content. And they gave us a statistically significant 1.6 bushels per acre, whereas we only have to have 0.7 of a bushel to break even. So they're both profitable. Then we get into these others that were just noise here, kind of that up and down. But then this one's a little bit different. We did have 1 site down in Southwest Michigan, very coarse textured, probably a loamy sand soil. I would say probably a Sphinx loamy sand, really low in organic matter and low in clay content. And that site actually reacted negatively to the Infero application. So I think we need to look at this more detail. But I think with our preliminary data here of these 7 sites, the products seem to perform well in those finer textured, a little higher organic matter soils than they do in the low organic matter, coarse textured soils. So that might be a take home there. We averaged all the sites together. We got 0.2 of a bushel. And that's not profitable across all the locations, but 2 out of 7 were profitable in the scenario that I described earlier. I want to thank several key partners here. First of all of our plot cooperators. 54 plot cooperators across the state of Michigan made these trials possible. The companies were just instrumental in giving us the product and really appreciate that. Bayer Crop Science, Corteva Agriscience, local seed dealers played a key role in applying the seed treatments at no additional charge for our trials. Syngenta, Valent, and Wilbur Ellis, thank you very much for your continued support.